It's a tough act to follow here. I just wanted to express my gratitude for the invitation to come here and take part in this absolutely fantastic event. And I'm excited to show you how we are using organoid technology in our quest to model intestinal development, where we use this as a tool to complement studies in vivo. So I'm fascinated by how organs form during development, how they can go through a phase of almost exponential growth. And then when the size of the organ has been reached, the tissue can transition into a state of homeostasis, where the level of cell loss has to be excruciatingly finely balanced with that of cell gain. And I believe that if we can understand at the molecular level the mechanisms that controls cells in these individual states, but also that are important for the transition from one state to the other, this is very likely to provide us with insights into what happens when we have to rebuild a tissue following severe damage, or potentially when we have the development of tumors. You heard from Hans that we have a grandmaster stem cell located at the bottom of crypts expressing LGR5 during steady-state homeostasis. But this is during steady-state homeostasis. I think one of the key questions that arose when I started my lab was, what about when we go outside homeostasis? What happens during development? Do we have cells obeying this exact same cellular hierarchy? What happens during regeneration? If we are reliant on a small subset of cells during regeneration, that might actually make us vulnerable to severe tissue damage. And then last year, we showed that following tissue damage in the colon, when the tissue is essentially completely lost, where we have a few scattered epithelial cells left, these undergo a very dramatic transition from their normal stem cell state into a fetal-like state, and we could demonstrate that this was orchestrated via excessive signaling, via the YAP-TAZ signaling pathway, where the cells were exposed to a complement of new extracellular matrix proteins in the context of a new growth factor environment, allowing these stem cells to potentially transition from their homeostatic state into a regenerative state with fetal-like, where the cells had fetal-like properties, driven by this excessive YAP signaling. And here we proposed that it's the stem cells that are capable of transitioning into this state. However, when we consider the enormous amount of papers that has been published over the last couple of years demonstrating that not only stem cells participate in regeneration, that also more differentiated cells can take part, one could speculate whether progenitors and potentially more differentiated cells are capable of undergoing the same transitions into this fetal-like state and thereby contribute to tissue regeneration. But this sort of poses the question, what is so special about this fetal-like state? Is cells in a fetal-like state intrinsically different to those you would normally find in a homeostatic state? So if we consider intestinal development, it starts out very early as a pseudostratified epithelium. So this is way before what you see here. And then at around embryonic day 14.5 in the mouse, this tissue transitions into a simple epithelium where all the epithelial cells align their nuclei along the basement membrane. Within a couple of days, tension builds up in the muscle. The buildup of tension in the muscle allow the epithelium to begin undulating and forming these rudimentary villus-like structures. And then in between these villi, you have an ocean of intervillus regions where cells are proliferating. And it's not until postnatal, the first postnatal week in the mouse, that you get the formation of the adult crypts, where you have cells expressing adult stem cell markers intercalated between Paneth cells. Interestingly, at E16.5, we have the same complement of proteins being expressed here within the intervillus compartment. And as you can see in this three-dimensional image here, you can see it forming a continuum in between all of these rudimentary villi. So I'm not going to take you through 4 years of excruciatingly quantitative fate mapping, but just give you the gist of this. So what we could demonstrate using quantitative fate mapping in combination with biophysical modeling in collaboration with Ben Simons at the University of Cambridge was that irrespective of whether you're sitting on top of one of these villi or within the bottom expressing adult stem cell markers, you have exactly the same probability to become an adult stem cell. And that what we could show was that remodeling of the epithelium is what governs this equipotency. That cells that are located on the top of these villi are capable of migrating into the intervillous region via tissue remodeling. And that cells sitting within this intervillous region, when we have the formation of the very first crypt structure, are the ones that are being selected as adult stem cells. So it's all about location. So can we somehow demonstrate that this equipotency is actually in existence? So if we now go back to intestinal development and consider these two structures, what characterizes these is that when we isolate cells from the adult epithelium and put them into matrigel with EGF noggin and aspondin, as Hans pointed out, we can get these beautiful budding organoids that we can maintain in culture for essentially eternity. However, what my lab showed some years ago was that if you do the same and isolate cells from the proximal part of the small intestine and put them into exactly the same culture conditions, rather than forming these budding organoids, what you get are spheres that can be maintained like spheres for years on end. And that these do not spontaneously transition into the adult counterpart, except when we add Wnt3a upon seeding, or when we transplant these cells into a colon where we've eliminated the natural epithelium. So this constitutes a way whereby we can then begin to assess how cells within this fetal epithelium, how these precursors to the adult stem cells are located. And this is where we come to the organoid part. We dissect, isolate single cells, sort out based on LGR5 CD44, which are located here at the bottom in this intervillous region, and the negative cells constitute the cells on the villi. If we culture these under normal conditions with E and R, what we see is that we get these beautiful spheres. And it doesn't matter whether these cells are sitting on the villus or sitting within the intervillous region. When we add Wnt3a to these cultures, irrespective of where you're coming from, you have the capacity to transition into the adult stem cell state to provide these, or to generate these budding organoids. If we isolate material from human fetuses at gestational week between 8 and 10, similarly, we can generate spheres from both villus and intervillous regions. And to challenge this, if we now take the cells cultured as spheres, expand them, and then subsequently infuses these into a colon where we've eliminated the natural epithelium, these cells are capable of establishing new stem cell niches, generate and give rise to all the differentiated offspring that you would normally anticipate. Meaning, in reality, that it doesn't matter where you are in the tissue. All the cells are equipotent. So what I hope this also has convinced you is that we have a stochastic selection of adult stem cells from an equipotent progenitor pool. This might be the reason why we, during regeneration, have to return to this fetal state, because all cells become even. And also that you have to be really careful when you look at marker expression, because marker expression is just marker expression. It is not providing you with necessarily a function. And that based on this, we believe that stem cell identity, at least in the intestinal epithelium, but we've also got evidence from other systems that this is an induced property. So what I hope I've been able to show is that cell fate decisions appear to be much more plastic, non-linear, as soon as we go out of homeostasis. That organoid cultures, I believe, can be used to complement studies of in vivo situations, where we now can begin to probe the molecular mechanisms that governs transitions between fetal and adult states, and that our preliminary data indicates that it could be super enhancer usage that dictates what you are and how your identity is regulated, as well as specific transcriptional regulators that might actually modify the overall chromatin structure. With this, I would like to thank my team, our collaborators, and our funding, as well as you for your attention. Thank you very much.